Hello and welcome to the Beyond Biotech podcast number 132. I'm Jim Cornell and this is the weekly podcast from Labiotech. Swedish-based company Anocca is at the forefront of TCR-T cell therapies and has a novel approach to T-cell biology, biotechnology and integrated software. To tell us about the company, its clinical trials and its partnerships is the company's CEO, Reagan Jarvis.
All right, so I guess to get things underway, I wonder if you could give us a bit of background on the company and its origins.
Yeah, so Anocca is a T-cell biotech, T-cell biology biotech. We're based in Sweden, and the company's built around a unique cell biology platform, which is built from the ground up to essentially address all the fundamental challenges across the discovery value chain for T-cell therapies. That includes target mapping, the generation of T-cell receptors, the targeting receptors of T-cells, and their characterization and optimization. We've deployed that, first of all, in deploying TCR-T cell therapies in the oncology space and built a very deep pipeline around that, and construct our own manufacturing facility into which we've deployed a gene editing manufacturing approach. First clinical program under regulatory review right now in Europe, which should be enrolling Q2 of this year. The origins of the company are a bit unusual in the sense that we're not an academic spin-out. This was literally an idea on paper, which was kind of a critique of how people were trying to develop products in the T-cell biology space. And so we've built the company from the ground up, including all the technologies and capabilities with a very talented and international team. So we've grown to about 130 people and over 40 nationalities represented in the team. So an exciting place.
You mentioned TCR-T cell therapy. I wonder if you could go into a bit of detail on what that is and the current level of use of that in treatments.
Yeah, a TCR-T cell therapy is basically programming a T cell to become a targeted therapeutic. So it's a cell therapy. What you're doing is programming these T cells with a T cell receptor, a TCR. And what they're doing is detecting native T cell targets, so HLA-restricted peptides. And these targets are derived from turnover of all the proteins inside cells getting chopped up and then loaded into these HLA complexes and presented at the cell surface. So this is what T cells normally do. They open up a window and look inside cells and see if there's targets they should react to, like mutated proteins in cancer or dysregulated proteins in cancer, or in virology setting, viral proteins. So TCR-T cell therapies in the oncology space are really about opening up a really powerful and tumor selective target space. So we're looking for things that are only expressed in tumors. And the caveat to that is that there's a lot of targets there and they're different between individuals because of the genetic diversity of tumors and the genetic diversity of individuals in their HLA genes. So the challenge really here is in scaling out your discovery, product development, and then clinical development. The current level of use is interesting. The first TCR-T cell therapy product was for oncology was approved just last year, a PharmaCell from Adaptimmune. And so that's a MAGE-A4 targeting product in the HLA-A201 HLA restriction in synovial sarcoma, a relatively niche indication, modest market opportunity. Behind that, we're seeing Immatics move forward a PRAME targeting product into melanoma phase 3 study. And then behind that, we've got a very small handful of companies in phase 1. And of course, we'll be joining that small group early this year.
Your website mentions your novel approach to T-cell biology and cutting-edge biotech and integrated software. I wonder if you could explain that a little.
Our approach is novel in the sense that we're approaching this from a cell biology lens. So it sounds kind of obvious that we're making T-cell therapies, so we have to look at the cell biology, but it is surprisingly differentiated in the sense that everything we do across the discovery workflow is a cell-based assay. We map these HLA-restricted peptide targets from any gene sequence. In a cell-based assay, we generate the TCRs and then characterize these in very sophisticated assays where we can combine engineered T cells with engineered target cells in very precise and reproducible ways. The biotechnologies behind that are essentially allowing us to construct the cells that go into the assays. So this is a molecular genetics, molecular biology toolkit so that we can very quickly, precisely, and cost effectively generate these large libraries of cells that we deploy into analyses. And then finally, on the software side of things, obviously a huge volume of quite bespoke data here to capture and analyze. So we've constructed a software platform which does just that, of course. It captures the data, does the bespoke bioinformatics in there, and allows us to visualize the results. And in addition to that, we've constructed that as a sort of an enterprise software built by us for us. So it does everything from laboratory protocol development and review to logistics, inventory, reagent ordering, all of the above, time management. So it's enabling us to scale out the discovery in a very cost-effective way and keep the standards of obviously laboratory execution, but also data collection and analysis to a very high standard.
What are you doing differently to other cell and gene therapy companies?
Well, I mean, it's that cell biology approach, sort of ground up approach to mapping targets first and foremost. So oftentimes you're seeing companies and academics for that matter work through primary specimens, whether they're grinding up tumors and looking for targets directly in that material, or looking at patient T cells and T cell receptors and trying to de-orphanize them and go back to a target and a discovery methodology. What we're doing from the ground up is saying, in the oncology space, what is the tumor-restricted genetic sequence? Because there's lots of NGS data out there across many tumor forms, across many patients. So we can take that as a jumping off point and simply map the available HLA-restricted peptide target space in those tumor-restricted gene sequences. And then downstream of that, we can systematically generate TCRs, characterize them, and even optimize those in a directed evolution platform. And the point of this is, again, everything's a cell-based assay, so we're reading out the real cell biology at the center of both target presentation and T-cell responsiveness, which allows us to select optimal target receptor pairs and optimize those to go into products. I would say that the key to this is scaling out your discovery and product development. First approved product last year in TCR-T. The modality works. It's a matter of scalability because of the highly segmented nature of the target space. You need to build libraries of these products and personalize them to both patient genetics and the tumor target specifics of their disease. So this is about scaling out discovery, scaling out manufacture, of course, product development, commercialization. And so this is at the heart of what we're doing is precision and scalability in all those sort of key points across that discovery product development value chain.
What kind of challenges does the approach that you're taking present?
There are a number of challenges. It's a complexity challenge. This is non-trivial in terms of biology, which we would say we've solubilized, we've solved that challenge in mapping the targets, making optimal receptors. Downstream of that, it's manufacturing. We're taking a gene-edited autologous manufacturing approach for our first programs. We'd say we've buttoned that pretty tight as well. And really beyond that is scalability, being able to do this again and again and again. And we've built the business systems and obviously the R&D systems to do that. I think the key challenge facing the field right now is funding, financing. So I'd say that's the number one challenge that I'd point to as the CEO at Anocca. But in terms of the science and product development, we think we've solubilized most of the challenges here. Looking beyond all that, really, it's about how do you build a commercial manufacturing, distribution, sales model behind these types of very advanced personalized therapies. That's, in a way, future problems. This is some years down the track, but we have our eye on that in terms of both the scalability of the manufacturing, the clinical immunology that you'll need to actually stratify patients for selection, inclusion, so on and so forth in future.
And with the programs that you're working on, what conditions or diseases are you looking to address?
Well, the first program is an oncology TCR-T program focused on pancreatic cancer. And the targets there will be mutant KRAS and various HLA restrictions. And a little bit differently to some of our peers in the space is that this is a very focused single indication approach into pancreatic cancer. Oftentimes you're seeing products against a certain target of an HLA restriction trialed in all comers across all oncology indications that have that target expression. We're kind of saying it's a precision modality. Let's do precision trials and focus down on to single indications that has a number of benefits. One, And first and foremost is you can design your clinical trial to integrate very closely with existing standard of care, which gives you an opportunity to have a homogenized as much as you can patient population, which allows some benefits in terms of manufacturing, timeliness, and standardization, and importantly allows you to interpret early stage data because it's not a range of indications with a range of prior therapies. That's a bit unique in terms of what we're setting up there. Maybe to clarify is we are approaching this as a library of products even in this first program. So we'll have up to 3 TCR-T products covering different KRAS mutations and different HLA restrictions in our first clinical program for pancreatic cancer.
Was there a specific reason for choosing that condition as something that you wanted to go after?
PDAC, or pancreatic ductal adenocarcinoma, is an area of very high unmet medical need. The 5-year survival here is about 10%. There are not many treatment options here. Patients often respond to the first-line chemo, but very quickly relapse. So median survival from diagnosis is in the order of 8-9 months. So very high unmet need. There are a number of reasons for choosing PDAC from a biology standpoint, from the quality of the mutant KRAS targets there, their homogeneity more or less across primary tumor and metastases, but also, frankly, the immediacy of feedback on those clinical programs. You're going to see your survival data fairly quickly. There are some attributes to the standard of care where we've been able to be quite smart how we're integrating our therapy there. So, high unmet need, potential to make a big impact, but hard-nosed both biology and commercial considerations to selecting that indication.
How do you define success in terms of what it means to the patient ultimately?
We're going in as necessitated as a second-line therapy. So this is one advantage we have. The patients aren't going through 5, 6, 7, 8 lines of therapy like you might see in lung cancer. That's not because of any other reason that there's no available prior lines of therapy as to what we're doing. So, it is a chance at an effective therapy. More generally, I would say one of the promise around TCR-T therapeutics is their potential for very high safety profile and low side effects. So, standard of care in a lot of these high unmet need conditions are very harsh cycles of chemotherapies, which are tough to take. A lot of the times patients opt out of them. They physically cannot tolerate any more chemo, and that's part of the patient management there. So TCR-Ts as a precision therapy have the potential to provide very, very high efficacy, very low side effect treatment options here. Having said that, we're not dreamy-eyed. These aren't magic bullets. There's a lot of work to do here with the TCR-T modality. And, we're looking, as most others in the oncology space, I'd say all others in the oncology space, at smart ways to combine our therapeutics with existing targeted agents and other targeted agents in development. Combo therapies are a feature of oncology today. They'll continue to be a feature, even more so moving forward. So that's part of our clinical strategy around PDAC, and we really want to dig into that indication, make these therapies work, and not just alone, in combination with other immuno-oncology strategies.
What is VIDAR-1?
VIDAR-1, it is the first clinical program. VIDAR is a Norse god, sort of calling out our Swedish origins here as a company. And that is a TCR-T oncology trial focused into pancreatic ductal adenocarcinoma. It'll be a multi-center trial here in Northern Europe. So 9 phase 1 centers, and looking to obviously establish safety and early signs of efficacy for our TCR-T products in that setting.
Could you tell me a bit about your partnerships with EmendoBio and Shinobi?
Yeah, I mentioned in the manufacturing background there that we're taking an autologous gene-edited approach to our first TCR-T products. That gene edit is conducted with a CRISPR-Cas system with EmendoBio to deploy. So essentially what we're doing is taking our TCR constructs and knocking them into the native TCR gene locus. So we're delivering our construct whilst disrupting half of the receptor, which has some benefits in terms of potency and consistency of your product, because you're getting rid of at least half of that endogenous receptor, and there's no competition between existing TCR and therapeutic TCR. It's also important in terms of that scalability I keep mentioning. A lot of approaches are still using across the cell therapy space viral vectors for integration of receptor constructs. And that has a challenge in terms of timeliness and cost to getting to new products first in human. It's very expensive, it's very time consuming to make a viral vector. With a gene edited approach, your CRISPR reagents are built to stock, they're universal. And what you're doing for a new product is simply providing a DNA template for integration, which is much quicker and much more cost effective to bring new products to humans. In terms of Shinobi, you asked about, this is the flip side of the manufacturing challenge. So Shinobi, Japanese US company, they're primarily an iPSC company, so induced pluripotent stem cells. We've partnered with them to explore iPSC derived alpha beta T cells as a universal off the shelf source of T cells for manufacturing. So we're working with Shinobi to bring their iPSC and T cell expertise with our T cell biology and TCR asset expertise to build a proof of concept around this scalable future manufacturing approach for T cell therapies.
You already mentioned scaling up, and you've also mentioned, again, from your website, creating off-the-shelf products. How are you kind of moving towards that and scaling up and the associated costs to keep those as low as possible?
In terms of scale up, I would say that the first challenge is scale out. To demonstrate the potential of the TCR-T modality, you need to access the target space, which you need to bring multiple products to bear. So part of that is the gene editing approach. Part of that is the way we manufacture these products. We have our in-house facility, we have in-house process development, we have in-house technology development teams that feed our process development. So we're working to a continuous improvement model, both on the biotechnology side. So what are the reagents, constructs, biological tools you can bring to bear, but also the equipment side of things. So there's real life engineering solutions to bring to bear there as well. So that's looking first and foremost to the scale out, and of course, keeping in mind that you do need to scale up each of those individual products in future. So that goes hand in hand with selecting the best biology and the best engineering solutions to bring that to bear in the medium term. In terms of the off-the-shelf products, I mentioned the collaboration with Shinobi. That would be our play at the moment to off-the-shelf manufacturing. That field has a number of, I'd say, biological and technical hurdles to overcome. It's difficult to scale iPSC differentiation. The characteristics of those cells need to be optimized, not just in terms of potency, but also stealthing those cells from the patient immune system. They'll be rejected just like most allografts over time, or acutely in some cases. So, Shinobi is a great team. They've got a great technology base, both on the iPSC and the stealthing from host immune system. And we're very optimistic to bring some proof of concept during this year to move that forward.
How do you see TCR-T therapy and the whole cell and gene therapy field evolving?
I'll stick to the T-cell biology lens there, maybe, rather than having a general riff on cell and gene, which is very broad. To address the target space, you need to bring many products to bear, and even in single indications. I think that is one major theme you'll see. Yet more generally speaking, if we say cell therapies and gene therapies, where you could say that case is vaccination and vaccination-like strategies where you try to provoke or suppress T-cell immunity in different disease settings. One of the sort of macros across the field is there are a lot of great manufacturing platforms. In the cell therapy space, a lot of investment behind CAR-T initially into ways of manufacturing, ways of enhancing your cells, and so on and so forth. A lot of manufacturing capability and technology out there. In the vaccine space, of course, we saw mRNA and LNP technologies come through, be validated during the SARS-CoV-2 pandemic, of course. And the theme really is there's a lot of capability to manufacture products, but the fundamental barrier to entry is what are you going to encode in those products? And that's really what Anocca has been built to address, is pulling apart the targets for T cells, and of course being able to provide T cell receptors for cell therapies, but more importantly, understand viral genomes, autoimmune targets, if you want to provide vaccination or tolerizing vaccine strategies. I think the theme you'll see is more emphasis placed on what to encode in these manufacturing platforms, which is, in a way, part of the overall trend in the industry where you see, modalities being able to manufacture before the real energy behind it. It's just unique in the T-cell biology space because the target space, whatever indication you look at, is segmented and requires that scalability I keep talking about.
I think one of the biggest buzzwords at the moment in the industry seems to be artificial intelligence. Is that impacting what you're doing?
The large language models, the generative AI, generally impacts all businesses to an extent. People are using those tools to search and summarize, which is what they're good at. In terms of clinical development, you're seeing a lot of value to add in clinical development in terms of patient population selection, the stratification, the downstream stuff there in clinical development. And that's owing to the fact that you've got really deep patient records, genomic data, and so on that you can crunch in machine learning models. In terms of discovery in the cell and gene therapy space, there's really limited use cases at this stage. And that's not to say there's not potential, that there certainly is. But the challenge with cell therapies in particular is that they're cells. It's very hard to model, even if you had very good quantitative, high parameterized data. Certainly our platform is designed to build that data. We're not there today. I don't think anyone really is. And that's a view on why we've put a lot of effort into our fundamental software platform, being able to capture that data, categorize it, and have that warehoused. So when we think we've got enough volume of data in specific use cases, we can start building those models on the cell therapy side. I would say there's one exception here. It's the target mapping. This is an unusually tractable area where the HLA-restricted peptides are formulaic in the way that they're loaded into certain HLAs. There are patterns and there are rules, loose rules in some cases, to that, and that you can quite well model which peptides will fit in which HLAs. And that's the basis, for example, of personalized cancer vaccines from BioNTech, just as an example. They're using computational models to predict which neoantigens, which cancer-specific targets should be encoded in this personalized vaccine on a patient-by-patient basis. I would say that more generally, if you're getting away from that fully individualized space, which we think you have to in the cell therapy setting just because of your relative complexity of the products to develop, that the number of products you have to bring to bear just don't really need that. If you've built a platform like we have to analyze the cell biology, you analyze the cell biology. You'll have to do it anyway. We're doing that in a scalable manner. You don't need those machine learning leg ups in the first instance. All that's to say that potential, but you need a lot more data. You need a lot more high quality data. I mean, I think that's a theme when a lot of people talk about AI. People often say rubbish in, rubbish out, and I think that's particularly the case for complex biology like the target T-cell interaction at the center of the modalities we work with.
You mentioned joining the party in 2025. I wonder if you could take us through the timeline of where you're going with trials and where the company's going.
Yeah, so this first program's under regulatory review here in Europe. So we'll see that all going well, start enrolling Q2 of this year. Would expect a phase 1 readout on the first product of that program second half of 2026 and accelerate that recruitment with activation of more sites to have a phase 2A readout in second half of 2027. So relatively modest patient numbers, of course, in the TCR-T setting. So, quite quick, relatively speaking, to get these trial readouts. More generally, I think what we're looking at is more programs because I keep saying it's scalability. We need to address this target space. We have a very deep pipeline of oncology assets and have some opportunities there. We're also looking very hard at non-oncology applications for TCR-T therapies. So we're looking to launch a second clinical program towards the end of next year, 2026, but not announcing what that will be, not even the disease area it will be in right now. This is the trajectory for the company is leveraging this discovery engine that we've generated to map targets, make assets, and of course the manufacturing capability we have to actually deploy those to human trials on a continuous basis. Certainly we're a platform biotech. It comes in and out of fashion, that expression, but we're looking to run platform biotech business model which is wholly owned programs to build proof of concept, build value around those products and the company, and partner in smart ways with pharma and big biotech to do discovery programs, product co-development, and so on, to further validate, obviously, the platform and spread the risk in some sense, but also just to execute on that business model, which frankly has been the only model to be successful as a platform biotech.
Great, thank you. Is there anything that you would like to add?
It's an exciting year for Anocca. We're entering the clinic for the first time as a company, and on the back of now 11 years of build-up of the company. We've put a lot of time and effort and obviously capital into building a discovery engine and our pipeline, of course, the manufacturing capabilities, and now it's where rubber hits the road. Developing products and trialing them in humans is what this industry is all about. And so it's an exciting time for the team. It is a great team. Scientists, obviously, engineers, quality management people. So yeah, looking forward to getting that done and seeing the results in the near future.
Definitely a company going places in the TCR-T therapy field. And the bottom line, of course, is hopefully making a positive difference for those suffering from pancreatic cancer. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and you'll join us next time for another Beyond Biotech.